Good afternoon, everybody. Thank you for joining us today to hear from my colleague, Dr. Wendy Francis. Wendy's going to present to you on employing genetics and genomics in healthcare. So without further ado, Wendy, over to you.
Thank you very much, Emma. So hi, everyone. Thank you very much for joining us. So as Emma's just introduced me, I am Dr. Wendy Francis, and I lecture and I contribute to numerous different modules across a whole range of undergraduate degree programs. So if you embark on your journey with us in September, October time, then no doubt you will see me at some point. Now, right at the beginning of your first year as you join us, there will be a genetics and evolution based lecture, and I lead on the genetics and evolution module, and I lead on the genetics and evolution module for applied medical sciences and medical pharmacology, but there is also a similar module that will be for the other undergraduate degree programs as well. So I'm just going to share my slides with you. There we go. Okay, so today's lecture will be on the genetics and genomics in healthcare. And the aims of the lecture really are to give you a bit of a taster lecture, and I'm going to do a basic introduction to genetics and genomics. And we're also going to have a little look about the application of using genetics and genomics in medicine and why it's important in medicine. We'll have a little look at the Human Genome Project because that really has revolutionized how we do or how we use genomics in terms of medicine and healthcare. And then we'll look at the impact of genomics on health and disease. So a good place to start is looking at the DNA. So we look at deoxyribonucleic acid. Okay, so DNA is essentially our blueprint. I'm sure you've all heard of it. I'm sure you must have all studied it perhaps in school at some point. And this is the sort of traditional icon of molecular biology. So I'm sure you're all familiar with it. So DNA is found in the nucleus of all of our nucleated cells. And we can see it's made up of two strands of DNA. We've got an outer sort of backbone, phosphate sugar backbone, and then inside the two strands of DNA are all the nucleotides, all the nitrogenous bases. And the structure of DNA is in like a helix. It's in a sort of spiral. So there are four nitrogenous bases, so these nucleotides, that complementary base pair in the center of the double DNA helix. There's adenine, which we represent with A, there's guanine, G, thymine, T, and cytosine, C. And we can see that adenine and guanine are this double ring structure, and thymine and cytosine are this single ring structure. And that's really important when we talk about complementary base pairs. So here's James Watson and Francis Crick. So they really put the structure of DNA together back in 1953. And they did that with the help of Rosalind Franklin. So she was producing some really clever images using X-ray diffraction of different protein structures. And one of those protein structures was DNA. And we can see here, this is an image of DNA. Basically, they just shine a light source through the DNA sample, and then the light refracts off of the protein structure onto a photographic plate. So James Watson and Francis Crick could identify that the DNA had two strands, and it was helical in shape with the sort of sugar phosphate backbone on the outside and then all the nucleotides within the middle. And they were also able to work out the sort of width of the double-stranded DNA helix as well. So they were able to identify that a double ring structure complementary base pairs with a single ring structure. And if we look at the DNA structure, here we've got two strands. It's not in a spiral, this is where it's been opened out. And you can see we've got one strand here and another strand here, and they run anti-parallel to each other. Okay, so this strand runs from the 5' to the 3' direction, and the other strand runs in the other direction. And basically what we mean by 5' to 3' is if we look at the number of carbons on the deoxyribose sugar. Okay, so we've got 1, 2, 3, 4, 5. Okay, so we've got the phosphate binding to the 5th carbon. Okay, and then the next phosphate on the 3rd carbon. So that's the 5' to the 3' direction. And we can also see that we've got guanine that complementary base pairs with cytosine, and we've got thymine that complementary base pairs with adenine. And this is always the case. So this complementary base pairing is really important because it's what enables DNA to replicate. So every time our cells divide by mitosis, the DNA has to replicate. So the fact we have complementary base pairing enables the 2 strands to act as a template for a new strand to be formed. It's also really important for DNA repair. So if there is some damage that happens to one of the strands, they can effectively use the other strand as a template to help repair it. So what exactly does DNA do? Well, most of you will know that DNA codes for proteins. As we said, it's the blueprint that codes for us in terms of how we develop and all the functions that our cells do. So along the length of our DNA, and we have a lot of DNA, we have these protein coding genes. So they're just sequences of nucleotides within our DNA double-stranded helix that code for a messenger RNA. So messenger RNA is a single-stranded molecule, and it's got nucleotides along it, okay? So basically, the 2 strands of the DNA will open up, and the messenger RNA will read off one of these strands of DNA. Now, in humans and in eukaryotic cells, this messenger RNA has to be modified. It's got to be processed. So there are elements within this messenger RNA that we don't want that need to be removed, okay? So these elements within this messenger RNA can be removed, then the mature messenger RNA copy can then move out of the nucleus of the cell into the cytoplasm, where it binds to a ribosome, okay? And the ribosomes are where protein production takes place. Now, proteins are phenomenally important to cells because proteins they are your building blocks to cells, and they're involved in a whole host of different functions. So this could be structural. So we could be looking at the cytoskeleton of the cell and the shape of the cell, or it could be involved in communication. So maybe there's protein signals that are coming into the cell, communicating from outside the cell. So these protein signals, they might bind to proteins on the surface of the cells. So these would be, these are called receptors. So we have the ligand binding to the receptor that activates that protein receptor. So we're transferring the communication from outside the cell membrane to inside the cell membrane. And then we've got like a relay system, if you like, where one protein activates another protein, okay, until we get down to the nucleus, for example, and then those proteins can activate within the nucleus, can then regulate the conformation of DNA, can open up the DNA, can regulate which genes need to be activated, which genes need to be switched off. So they have a regulatory component as well. Proteins can also be catalytic. I'm sure you've all heard of enzymes, and they can also be involved in transport as well. And transport is really important because there are some molecules that need a protein to be able to transport them around the cell. And we've got lots of different organelles within a cell that all need to communicate with each other. And they can do this through protein carriers. So I just wanted to have a little sort of delve a little bit deeper into protein production, otherwise known as translation. So our mature messenger RNA copy has moved out of the nucleus into the cytoplasm of the cell, and it binds to the ribosome. So we have the largest subunit of the ribosome and the smaller subunit of the ribosome basically clamping down on that messenger RNA molecule. So how then does it form that polypeptide chain or that protein? Well, this is our double-stranded DNA molecule. Okay, and we can see that the messenger RNA is transcribed, so it's copied from one strand of the DNA. And on the messenger RNA copy, we have a series of different nucleotides. And 3 nucleotides forms a codon, it's a triplet, okay? And one single codon of 3 nucleotides codes for a specific amino acid. Okay, so we can see that UGG codes for tryptophan, which is an amino acid, and UUU codes for phenylalanine, which is a different type of amino acid. And we've got a whole series of different amino acids that can make up a polypeptide chain. So basically, as the messenger RNA moves through the ribosome, these funky little RNA molecules here, they're called transfer RNA, they specifically bind to these triplets, to these codons within the messenger RNA. And then these transfer RNA molecules also bind to specific amino acids. So you can see that they bring in specific amino acids to that messenger RNA. So each tRNA comes in specifically binding to the right nucleotide triplet. And then the peptide bonds are formed between the different amino acids to form that polypeptide chain. Okay, so 3 nucleotides forms a codon, which codes for one single amino acid. Now, can you think what would happen then if we were to change one of these bases, one of these nucleotides within the DNA? Okay, if we were to change one of the nucleotides within the DNA, it has the potential to change the messenger RNA copy, which would change the codon and may, not always, may alter the amino acid it codes for. So sometimes we can get mutations that happen in our DNA, and that can, not always, but it can have an influence on the amino acid it codes for. And if that change in that amino acid significantly leads to a change in the protein structure and the protein function, then that has the potential to be pathological. It has the potential to be disease causing. So this is one little example to try and illustrate that. So sickle cell disease is one of the rare diseases that's actually caused by a single base change within the DNA. So this is our wild type beta globin gene. So here we've got our DNA, which is CTT, so cytosine, thymine, thymine. That then will code for messenger RNA, which ultimately codes for the amino acid glutamic acid. Now, hemoglobin is made of 2 alpha globin chains and 2 beta globin chains, okay? And it's what carries oxygen within our red blood cells. So these hemoglobin molecules, they reversibly bind to oxygen, and as the red blood cells travel around the body, they deliver oxygen to all the different tissues of the body. Now, in sickle cell, we have a single base change, and it happens in the second position of the codon. So instead of a T, we have an A. Now that changes the codon within the messenger RNA, which then changes the amino acid it codes for. So now we've got valine instead of glutamic acid. Now the problem here is that valine is hydrophobic. It doesn't like water, whereas glutamic acid does. So valine will do everything within its power to get away from water. Okay, so when the hemoglobin gives up the oxygen, or if you go into conditions or environments where there is low oxygen, valine will basically want to stick to something. It needs to get away from the water. So what you get is you get an aggregation of the beta globin, of the sickle globin molecules. Okay, so, and that will distort the shape of the red blood cell. So you get this sickle red blood cell, which can cause all sorts of problems because it can lead to blockages of various blood vessels. So that's one example where a single base change within the DNA can lead to a significant alteration in the protein structure and function to cause disease. So what else does our DNA do? Well, we've got various different protein coding genes across all of the DNA that we have. And in between those protein coding genes, then we have non-coding DNA. And to be honest, there is non-coding DNA actually within the protein coding genes as well, just to confuse you. But in our non-coding DNA that we find between the protein coding genes, there are areas that can be transcribed into RNA. Okay, and RNA, ribonucleic acid, are single-stranded molecules, and that gives them versatility. So this is ribosomal RNA. Now, ribosomal RNA is one of the largest RNA molecules that we have within our bodies. And you can see, because they're single-stranded, it means that they can complementary base pair to themselves. So they form these really big, elaborate structures. And because they can form these big, elaborate structures, that means that they can have a multitude of different functions. So in the case of ribosomal RNA, this has a structural component. So it's a structural component of ribosomes, okay, where protein production, where translation takes place. It can also have a catalytic function. So in ribosomal RNA, it can form the peptide bonds between the different amino acids that makes up the polypeptide chain. Okay, but there's a whole host of different RNAs that can be transcribed from the DNA. So some of these have a regulatory role. They can be really, really small. They're called microRNAs, and they can be involved in regulation of protein expression. Okay. There are also a whole host of different regulatory sequences, and proteins will recognize and bind to these regulatory sequences to regulate protein production and protein expression. So again, if we have a DNA sequence change within these non-coding DNA sort of regions, if they fall particularly within a regulatory region, that can significantly impact on protein structure, protein function, protein expression, etc. So I think it's really important to understand that mistakes are made in our DNA all the time, okay? These can be mistakes that just happen as a part of DNA replication. And we know a lot of cells are actively undergoing mitosis all the time, so the DNA has to replicate, and mistakes are made. We can also have various different types of DNA damage. These can be intracellular based, just from the simple act of metabolism and the products and the reactive oxygen species that are produced as a result of that metabolism. Okay, they can get into the DNA, they can damage the DNA. Or it could be environmental. So, I don't know, UV radiation from the sun, for example, or any chemicals we come into contact with. But we don't get ill all the time. And that's because the body has these DNA repair mechanisms. So DNA repair mechanisms are essentially proteins. Again, I told you proteins do everything. So these are proteins that can scan along the DNA and can pick up these various different damage that happens to the DNA. And they can repair it, or they can facilitate it by different proteins. But of course, because they're proteins, that means there could be a DNA sequence change that impacts on the DNA repair proteins. So perhaps the DNA repair proteins become dysfunctional. And if the DNA repair proteins are dysfunctional and they're not able to detect the sort of damage that happens to the DNA on a daily basis, then we've got a significantly higher risk of building up these mutations, these changes within our base DNA sequence, particularly as we get older. So that can ultimately lead to disease susceptibility, particularly things like cancer, for example. If we get a buildup of mutations that happen within our genome, within our DNA, that can lead us to disease susceptibility and developing cancers. So any undetected or unrepaired mistakes can be left as permanent changes within the DNA. But very rarely do these mistakes actually cause disease susceptibility, and that's because our nuclear genome is quite large. Okay, and the chances of these mistakes, of these mutations happening in an area of the DNA that's going to have a significant impact on protein structure and protein function is fairly rare. So when we look at genetics, we're looking at a very small section of the DNA. So that might be a single nucleotide change, or maybe a little string of nucleotides within that DNA sequence, okay, that perhaps changed, maybe have some kind of pathological input. When we're talking about the genome, we're talking about all of our DNA. And we can see that when we do a karyotype. So DNA is packaged into chromosomes. So these chromosomes, they could be long and stringy and really pulled out, or they could be really short and stubby like they are here. So this happens at a point during mitosis when the DNA is replicated, and we can see little short stubby chromosomes, which makes them visual. We can see them. It's a visual map of the genome. So we can see that we have 22 autosomes, and then we have our sex chromosomes, X and Y. And we get one chromosome from the pair from our mum and one chromosome from the pair from our dad. And it's a 50-50 chance which one we're going to inherit from which parent within each pair. So looking at this karyotype then, can you tell me if it's male or female? It's a rhetorical question. You don't have to put it in the chat. So yeah, this is a female karyotype, okay? So we've got 2 X chromosomes. If it was a male karyotype, it would have 1 X chromosome and then a tiny little Y chromosome. Okay, so this is a female karyotype. And if we look at these paired chromosomes, we can see that they're the same length. They've got the same centromere position. So the centromere is usually, you can visualize it as a little pinch point on the chromosome. So here we can see it. Think of a belt being pulled really tightly around the chromosome, and it's where the sort of pairs anchored together. So here we've got another centromere. I think it's here, and you can see it's quite high on chromosome 4 and chromosome 5. We also have a long arm and a short arm of the chromosome, and hopefully you can also see a similar banding pattern between the 2 chromosomes within the pair. But I think it's important to remember that, of course, we've inherited 1 chromosome from the pair from our mum and 1 from our dad. So the DNA sequence is not going to be identical. The banding pattern and all those gene loci, all those different protein coding genes is going to be there and identical, but the actual DNA sequence between the 2 chromosomes is not identical. So karyotypes can be really useful, and we can identify some genomic abnormalities from a karyotype. So if you have a look at this karyotype here, can you try and identify what the genetic abnormality might be? You can all pretend to be cytogeneticists now. So hopefully, looking at this, you've established that it's male. It's a male karyotype. We've got an X and a Y. But did you pick up on chromosome 21? So chromosome 21, you can see we've got 3 chromosomes, and this is known as trisomy 21, which is otherwise known as Down syndrome. So you can see that we can use the karyotype for identifying some genomic abnormalities. This would have happened, incidentally, if you're interested, because during meiosis in the development of the egg cells, it's usually within the mother, in the development of the egg cells, you've got what they call meiotic non-disjunction. Basically, you've got too many chromosomes going into one gamete and not enough going into the other. So the woman's egg would have had 2 chromosomes, and then of course when it fertilized with the sperm, it brings in the other chromosome 21, so we end up with 3. So you can identify structural abnormalities from a karyotype, so we can look at abnormal banding pattern. There's also certain translocations that you might be able to detect. So that's a rearrangement when we're moving, sort of rearranging genetic segments from one chromosome to another chromosome. Okay, that's called translocation, and there are translocations in leukemias and cancers. But essentially, when we're looking at a karyotype, we can see it's fairly low resolution. You can't see the DNA sequence, and really you're only going to be picking up sort of larger structural changes within the DNA. And that's really where the Human Genome Project comes in. So the Human Genome Project really has revolutionized our ability to be able to diagnose and to treat disease. It was a particularly ambitious project. It was run in the 1990s. It cost Lord knows how much money to do. But basically, the Human Genome Project sequenced the human genome. And when we talk about sequencing the human genome, basically, we're just trying to find the order of the nucleotides within the genome. And when they've sequenced the genome, they determined that the genome is actually over 3 billion base pairs in size. It's pretty big. Okay, and I've actually written it in a number as well, because I think that's a difficult figure to try and visualize. So it's 9 zeros. So that's a tremendous amount of nucleotides within our genome. Can you imagine having your genome sequenced and then someone hands you a book? You know, there you go, there's your genome, there's all your nucleotides in order. You just go through it page by page of G, C, A, and T. Okay, I mean, it wouldn't mean anything, would it? So now there is a science called bioinformatics, which uses different computational tools and different computer software programs to try and understand exactly what all that data means. Okay, so if you have a love of computers and you have a love of biological science, this is a great way of combining the two. So by doing the Human Genome Project, they determined that only 1%-ish of our genome codes for proteins. And if you think about the wealth of different functions that proteins have within the cells, they are our building blocks. That's quite incredible that only 1% of our genome actually codes for proteins. And that 1% equates with around about 20,000 to 25,000 protein coding genes. Okay, but we know there are 100,000 plus different proteins functioning within the cells of our body. So you'll have to come on the degree course to find out how we get 20,000 protein coding genes into 100,000 different types of protein. But what the Human Genome Project did was it provided us with a reference genome. And this wasn't worked out from one individual, it was worked out from multiple different individuals, but it gives us a representation of what the genome is that we can refer back to and we can compare to. So there are elements of our genome that have high variability. So that would be represented by multiple different representations showing that high level of variability. So when we sequence the genome now, what we can do is we can use this reference genome to map to, and we can identify changes or differences to that reference genome. And any changes, any differences that we determine from any genomes that we now sequence that are different to the reference genome, we can call them variants. Okay, so they are DNA variants, they are different to the reference genome. So the Genomics England, there's a whole host of information on Genomics England if you want to read more about this, and it's really well explained. This is one of the fun facts that the Genomics England website put out. So your genome is only around 0.1% different from any other person's. So the person you pass in the street, for example, or that you meet at the supermarket, your genome is only 0.1% different. Again, that's quite incredible when you look at us all, and we don't look the same. We're all very unique. Okay, but actually, because our genome is over 3 billion base pairs long, that actually equates to 3 million differences in our DNA. And that's really important because that really does give us our individuality, our hair color, our eye color, our blood type, our characteristics. Okay. But variation, genetic variation, is very important. It can contribute to health. So changes within our base DNA sequence, as I said, it's not always pathological. Sometimes it has no effect whatsoever on our phenotype, so no biological effect. Or sometimes it can contribute to health. It can contribute to height or personality. And these are all factors that are also heavily influenced by the environment, by the way we've been brought up, and by what we eat, for example. Genetic variation is also really important because it's what enables species to adapt to changes in environment. So perhaps if we take the example of a bird, and that bird has had a genetic variation that has occurred, and maybe that genetic variation has led to a change in beak shape. So suddenly they can enjoy a food source that they couldn't previously access. Okay, so that means that that bird will likely thrive. Now, if they can pass that gene down to their offspring, okay, that means that offspring will also be able to eat that particular food source and thrive within that environment. And that enables species to change or adapt to changes in their environment. Excuse me. So sometimes genetic variation can be good, but other times genetic variation can be bad, and it can lead to pathological change. It can lead to disease susceptibility. And the important thing is identifying which variations, so which changes within our DNA sequence are completely normal and contribute to health, and which are potentially pathological. Okay, and that's really important because we don't want to be identifying variations, so differences in relation to the reference genome that actually have no pathological connotation at all, okay, or no disease susceptibility. So as technology has advanced and the cost of sequencing has come down, we've been able to do much bigger projects. So this was a project that actually completed right at the end of 2018. I use the word completed advisedly. So this was the 100,000 Genomes Project, and it generated a whole wealth of data, which they're now obviously still analysing. And the reason why this was possible was largely because of this machine. It's called the Illumina sequencing machine. So the aims of the 100,000 Genome Project was to sequence 100,000 genomes from 85,000 NHS patients with rare and inherited diseases or cancer. And not just the patients, but also maybe their parents as well, because we wanted to have a look to see if we could establish or find the same variations in the patients as the parents. So is it sort of an inherited genetic disease, or has it happened within the individual during development, for example? They found actionable findings for this. 1 in 4 rare disease patients would have gone on to get a diagnosis or have the potential for therapy, perhaps. And in 50% of cancer cases, they had the potential for therapy or clinical trials. So quite a lot of people have actually benefited from this 100,000 Genomes Project. And I know you can't follow a link very easily on a webinar, but if you just Google participant stories, Genomics England, there are some lovely little videos on the Genomics England website that you can watch. And it shows you individuals who have benefited from the 100,000 Genomes Project. So perhaps they've gone through years of testing and not come up with a diagnosis, but then following the 100,000 Genomes Project, they've been able to identify the variation within the DNA and establish the dysfunctional protein, for example, or to be able to give that patient a diagnosis. So there is a significant benefit to healthcare by being able to sequence our genome. You can't get your genome sequenced on the NHS at the moment unless you are involved in one of these projects like the 100,000 Genomes Project, okay? But the potential is there to massively improve diagnostics by identifying these variants that are linked to rare disease or cancer. Sometimes diseases are caused by variants in one particular gene. So if you have variation in that gene, then you are susceptible to developing that particular disease. Other times it's not so clear-cut. So we might have a whole range of different variations within the DNA sequence in multiple different genes. And maybe that DNA variation in itself might have no effect on your characteristics, on your phenotype, we call it. But collectively, multiple different variations across multiple different places within our genome might work together to create a disease. And there are some diseases like coronary heart disease, or maybe diabetes, type 2 diabetes, where there are multiple different variations across the genome. And if we can identify those variations within the genome, then we might be able to come up with sort of a predictive risk score. And this is where predictive comes in. And this is where sequencing the genome might be very beneficial to healthcare, because if we can identify these variations, and we know these variations are associated with the development of a particular disease or susceptibility to disease, then we would be able to put certain intervention measures in place. So maybe the patient would be able to change their lifestyle accordingly, or perhaps put some kind of medical intervention or therapy in place. And of course, if they pick up on certain variations within our genome that lead to disease susceptibility, for example, cancer, then we would be able to put increased screening in place to try and identify that disease development much earlier. There are, of course, people like Angelina Jolie who have, they detected the BRCA1 mutation. So she was able to put quite invasive measures in place to prevent the cancer formation. So BRCA1 mutation is one of the DNA repair proteins, codes for a DNA repair protein. So you can get a buildup of genetic mutations. So just because you've got the BRCA1 mutation doesn't necessarily mean you're going to develop ovarian or breast cancer, but it does certainly increase your risk of developing that particular disease. So it just gives you that option of putting those predictive measures in place. Also, benefit to healthcare is pharmacogenomics, which is a really posh term for basically saying how drugs interact with our genome, or the role of the genome in drug response. So this can facilitate personalized treatment. I don't know if you've ever thought about it, but have you ever wondered why maybe people on the same drug suffer different side effects that could have a genetic component? Or perhaps you have two people with outwardly looking exactly the same tumor. One tumor responds really well to the drug, the other one doesn't. Okay, so you haven't got that one drug fits all anymore. So an example of that would be in breast cancer again. So some people with breast cancer respond really well to Herceptin and others don't. And the ones that really respond well to Herceptin are the ones that are positive for the HER2 gene. Okay, so the genes you express can influence the way that drugs work on those particular tumors or those particular disease. Also in terms of identifying new drug targets as well. And we've seen in the current COVID-19 pandemic, of course, that we are sequencing the virus and we can look for different variants. I'm sure you've seen it on the news where we can identify different variants of the virus and we can track those viral strains. And there were mobile sequencing laboratories out in Africa during the Ebola crisis as well, doing a similar idea, trying to track the different variants of the viral strain. But also, in addition to healthcare, I think we can't overlook the importance of looking at genomics in terms of research. So there are people looking at evolution, and it can also be really helpful for forensics as well. So I just wanted to finish with this little picture. So this is the Singleton campus at Swansea University. You can see we're right on the beach, and I feel very lucky to work on the Singleton campus within Swansea University. This building here is the Institute of Life Science 1, so this is where I work, and a lot of the research within the medical school at Swansea University takes place in the Institute of Life Science. And you can see here is Singleton Hospital. So we've got direct links to the hospital from the university. And of course, Morriston Hospital is not far away either. So there's good potential for collaboration as well. Thank you very much.
Fantastic. Thank you so much, Wendy. Really informative. I've been learning so much attending and hosting all of these lectures. Really informative. So those of you that are watching this on record, just to say, because you can't participate in the Q&A, if you do have specific questions, then you can email us. It'll come through to the team. The email address to use is study@swansea.ac.uk, and we'll do our best to come back to you and can liaise with Wendy directly if you've got specific questions about what we've covered today. So I'm just going to end the recording. So thank you for watching the session.